Thank you for the nice invitation. It's a great pleasure to be here, and so far I have learned a lot. It was a very nice interaction. Today I would like to review kind of a long series of studies that many people conducted, which I would like to thank first. These are people that are linked to the projects I'm presenting today, and I will mention their contribution during the lecture further. Moreover, all my research is funded by taxpayers' money, so thank you for paying your taxes. And that money was distributed by these organizations so that I could use it. After 4 days of the winter school, I thought some more active part would be important to have. And therefore, I would like to use you as subject in a brief experiment. First, as an kind of an introduction, of course, I don't have ethical approval. I don't have informed consent. So if you don't like to participate, you have to close your eyes for a few seconds. Because if you keep the eyes open, I will change your brain, at least for a few hours, perhaps for a few months, weeks, perhaps for the rest of your life. So it's quite a massive brain manipulation. And so please, if you don't like to take part, please close your eyes now. So you have seen a picture of those that kept the eyes open. And I'm quite convinced that you will be able, at the end of even 2 lectures, I never did that with 2 lectures, you will still be able to recognize that face. And so somehow, within these 1 or 2 seconds I showed you the face, something changed in your brain for hours, perhaps your lifelong. So this process is the kind of my personal aim in research. How do we do that? That we are able to see this, and then afterwards our brain is changed so that we can recognize the picture. During the week, listening to different lectures, I got the impression that the hippocampus, kind of the core structure of our brain, is kind of a video recorder of life. It just records everything that comes to us. And I would like to argue that it doesn't. There are many, and I think it was also clear in most of the lectures given, but there are many reasons why it isn't. It's, of course, biologically not plausible. It's not our experience that we can remember everything, and we have even heard that remembering everything is kind of disturbing our life. And we have heard a lot about the capacity about the brain, how many memories can we store, and somehow the agreement came down that the hippocampus is quite limited. The neocortex appears to be unlimited almost. And if you just think about how much information we perceive with our senses, so this is just an estimate based on bits per second. And so the older of you just remember the newer floppy disks, the hard ones, the smaller ones, had 1.4 megabytes. So per second, one of those floppy disks. Nowadays, that's of course not much anymore. But still, in an 80-year life with 8 hours sleep per day, we end up with a 2,500 terabytes. So we don't get everything in. But what determines whether we remember an event or not? Just imagine that you have watched the news last night, and today, if I would ask you what were the news items, you may be able to remember some of the items, but others you may have forgotten. So why did you remember some, and why did you forget others? So there might be just a disturbance in the perception. There was some noise and you couldn't hear what the speaker was saying, or somebody was just crossing between you and the television and you couldn't see it, or your attention slipped. Yeah? So you didn't pay attention, you were thinking about something else, and therefore you couldn't remember. But there might be also kind of some news that might be more emotional and arousing to you, and therefore you remember them better than others. There might be also some other news where you have some contextual information. You can kind of bring the news item into the context of your knowledge, and you're able better to remember that because you do these semantic associations while you are watching the news item. So you remember that item better compared to other news items where you have no clue what they're talking about. Or whether very unexpected novel news items might also be better remembered than the ones you already expect. But since the patient HM and the careful investigation and very innovative investigation of that patient by Brenda Milner let us know that there is an additional process that might be disturbed. It's just specifically memory. So patient HM had no problems with perception, no problems with attention, no problems with emotional processing or semantic processing, and also his novelty detection was probably normal, although I'm not sure whether it was formally tested. But the memory was still heavily impaired. He was unable to remember things he has encountered after the removal of both miotemporal lobes. In addition, he was unable to remember things that happened shortly before surgery. This careful investigation led not only to the conclusion that there is a specific brain region that is exclusively related to memory, and that there's something like a specific brain process memory, not just a side product of all these other processes just mentioned. And furthermore, Brenda Milner and others investigated patient HM and other miotemporal lobe patients so carefully that a whole taxonomy of different types of memory was developed. And you have all seen that. That's a textbook knowledge nowadays. I'm mainly interested in the declarative memory, so memories that you can declare, you have conscious access to them, and you can talk about them. And usually there are two types defined: the episodic, very often discussed now in this meeting, the ability to remember a certain event, what, where, and when that happened. Semantics is already a bit less well defined, semantic memory. I'm working in an institute that is dominated by linguists, and when I use the term semantic memory, kind of they turn already their eyes. So we are, in the memory field, we are very poor in defining that type of memory. Usually we define it as word knowledge or as episodic memory minus the episodic part. So without the access to when and where you have learned that, there's the idea that you have learned somehow, somewhere, somewhen, where the Eiffel Tower and what the Eiffel Tower is, but you don't remember anymore where you have learned that. And then that's semantic memory. Huh? It's just the episodic memory minus. And it's even unclear in the field, I think, whether you first form a semantic memory and then you add the episodic details to it, or vice versa. You have first an episodic memory and then somehow the episodic details vanish. So at that corner of the nomenclature of memory, I focus my talk, and all these kinds of forms of memory I ignore. In addition to the different types of memory, there are also different processes, and we have heard them already very often during this week. So there is a process like encoding, so the transformation of perceptual or thought input into a memory. I call it memory formation. And later on there's a retrieval when you remember something, when you recognize something. Something might also happen in between, but we will come later to that. First, I would like to only go into memory formation. Actually, I will hardly talk about retrieval. If I have time, if I'm too fast, then I will have some reserved slides on retrieval. Investigating memory formation was for quite a long time relatively difficult compared to investigating retrieval, because retrieval is very nicely time-locked. You can make a recognition memory task and you know exactly when somebody is recognizing a certain item or not. That's easy for the analysis. But there's also a way to study the encoding part. And two studies by Jim Brewer in John Garbiedi's lab and Anthony Wagner in Randy Buckner's lab at that time, they published already in 1998 two science papers back to back using the same procedure. I think Roberto already mentioned the procedure briefly, but I would like to go a bit more in detail in that procedure. It's a so-called subsequent memory effect or subsequent memory procedure, or the M effect. For those who are not in the field of cognitive neuroscience, they might not know that. So therefore, I would like to explain that carefully. You have a training, learning, or study phase where single items are presented one after the other. These can be words, pictures, whatever you like. And you record brain activity either by ERPs. Actually, this approach was first initially developed for ERPs, for event-related potentials. And then you have many different items, and you record brain activity associated or time-locked to these events over and over again. And then after a while, you have a test phase. All kinds of different memory tests you can do, free recall, acute recall, recognition, whatever you like. Let's assume it's a free recall. And then the subject mentions names that he or she has seen during the study phase. And then, for example, when he says the word milk, you can code the encoding activity as subsequently remembered. And you can do that for all the words the subject has mentioned. And then you can average those, and you can contrast that with the average of all the other trials. And then you get a difference in brain activity between later remembered and later forgotten. It's kind of a predictive activity of which items will be later remembered or not, based on post hoc sorting, based on the test performance. When you do that with fMRI, and as Jim Brewer and Anthony Wagner have done very nicely, and at that time it was very difficult to do event-related potential. I published a paper in a similar time where I wasn't able with our machine and technology to do that. And for example, in Jim Brewer's study, they used real-world photographs of landscape pictures or indoor pictures, and they could do nicely the subsequent memory effects on the right. There's just this picture processing, and you see in the medial temporal lobe, quite posterior, more parahippocampal than hippocampal, you see activity that was stronger for later remembered as compared to later forgotten items. This is just a single subject division. So you can also, of course, do kind of a time course of the fMRI activity. You see the scan here. We are here the number of scans, and you can see that the later remembered and familiar and forgotten, as they called them, it's not really familiar, I would say, but you see that there is an effect in the right frontal cortex, left parahippocampal, right parahippocampal, that was larger for later remembered than forgotten. Anthony Wagner used words, and he has again, now on the left prefrontal cortex, a nice difference due to memory in the more anterior part, but also in the parahippocampal fusiform on both sides. Two control regions in the visual cortex and motor cortex don't show the subsequent memory effect. So these results are extremely robust. With ERPs, doing in the subsequent memory effect is extremely difficult. And with fMRI, somehow it appears to be very simple, and nobody really understands why that is. The subsequent memory effect, now there are hundreds and hundreds of studies using a subsequent memory effect. The surprising result in this paper, or these two papers, was actually that the subsequent memory effect was in the posterior parahippocampal cortex fusiform gyrus, and not in the hippocampus. Everybody was expecting entorhinal, perirhinal, and hippocampus. But they didn't detect that, at least not at that time. At that time, it was very difficult to get good fMRI signal from the medial temporal lobe because there are large artifacts due to air-filled cavities, and therefore they had really trouble to get that. In addition, of course, fMRI has not a good temporal resolution, and therefore I went on at that time and tried to investigate that subsequent memory effect with electrophysiology. And similar to the single cell recording presented on the second day or first day, I don't know, source memory. We used at that time also intercranial recordings, but normal field potentials, normal large macroscopic electrodes. We used the design as shown before, wordless learning, a distraction task, and a test, and we had many lists So there were many circles. In total, the patients had to learn 240 words.
But the learning phase is just a sequential presentation of the words?
Each word is presented once, and they were instructed that they will be tested afterwards by free recall.
Free recall?
Yep.
How long are the lists? Does that matter much? Or?
The free recall?
No, how long are the individual lists? Because you said 200.
These are 12 words, so 20 lists. Yep. And these were high-frequency words, common words, and the patients were well able to do that. We investigated in that study 12 patients, and we took only patients that received electrodes bilaterally due to clinical needs. And we excluded all those patients, these were only a few, that did not get seizure-free after surgery. That means we took only those patients that had presumably one normal hippocampus. And we took only the data from that hippocampus. So the hippocampus contralateral to the side that was operated. So therefore, we had ERP activity or EEG activity from a more or less healthy medial lobe. What did we observe? So just the electrodes first. The electrodes were implanted from the back of the head. These were long electrodes with 10 contacts. Some of them were in the hippocampus, some of which were in the, as I called, rhinal cortex, because with that method we are unable to distinguish between ento and perirhinal cortex, where the signal is coming from. This is just a picture, it's hard to see, but here you see the artifact of the electrode. That's larger than the actual electrode. The electrode has a diameter of 1 millimeter. This is just a picture from the operating room. And this is a boy with way more electrodes during the daily checkup of the insertions, and these people have to wait for 1 to 3 weeks until they have enough seizures recorded. And during that time, they're often happy to participate in such an experiment. So these are the results, just to walk you through. This is the average, this is single subject data. What you see here is the time course. These are about 400 milliseconds after the onset of the word. What you can see is a negative, up is negative deflection of the potential, which is larger for later remembered than for forgotten. If you do kind of statistics where this effect is significant, it's about around 400 milliseconds. This is in the anterior parahippocampal gyrus, which is covered by the rhinal cortex. A second effect was observed inside the hippocampus, which started later and which lasted until the end of the stimulus presentation. So a long-lasting positivity. The difference between positivity and negativity here is irrelevant. Only deviation from zero is relevant, because the polarity depends on the spatial relationship between the electrode and the neural cell layer. So this one is now recorded with a depth electrode inside the rhinal cortex in the white matter, so you get a negativity. If you record the same potential with a strip electrode on the surface, you get a positivity. So orientation, polarity is irrelevant. This is just single subject data. The interesting thing is also that these two subsequent memory effects in the hippocampus and in the rhinal cortex are correlated across subject. These are the 12 subjects, and as you can see is those individuals that have a large subsequent memory effect in the rhinal cortex have also a large one in the hippocampus and vice versa. So somehow these two processes, sequential in nature, appear to be related. In the next study, we were interested, or actually we used the same data, but we analyzed it in a different way, how these two processes are related to each other. Guillen Fernandez, a postdoc in my group at that time, was very much interested in high-frequency oscillations. And the idea is that the communication between two neural areas, like the rhinal cortex and hippocampus, which are well connected, the main input structure to the hippocampus via the perforant path, is established by a synchronization, a phase synchronization of high frequency, so-called gamma. And therefore we asked the question whether the synchronization between these two structures is higher for those events that were later remembered as opposed to those that were later forgotten. And that's exactly what we found. This is a time frequency plot. So here's time point 0, where the word was presented, and this is the presentation time. These are the different frequencies investigated. At that time, gamma was around 40 hertz. Now it has changed a bit. What you can see here is very early on, there was a highly significant increase in synchronization, up to 30% increase for those items that were later remembered as opposed to forgotten. Later, at around 1,000 milliseconds and later, there was an opposite effect. There was a desynchronization between the two structures. Our idea was that it was a very early, I think I was very interested to see, I think you had the time course, how long it takes up to reach the hippocampus. That's even earlier, I think. We speculated in the discussion after the referee followed us up on that early onset that there might be some direct connections preparing from the LGN to the medial lobe, preparing or informing the hippocampus that there might be a salient stimuli, and that process might be sometimes more effective than other times during the presentation, the sequential presentation. This desynchronization later on might enable the hippocampus to end the process of encoding of this specific event and thereby protecting it for further input and making a clear memory. That was at least the interpretation we had at that time. So going back to this very simple graph, there's something in between. It's usually the word consolidation. This term, I think, is extremely poorly defined, and I would like to explain why. So, forgetting is often seen as something different than consolidation, not always. Might be sometimes only a byproduct of consolidation. The stabilization, actually, everybody kind of links to consolidation. Perhaps also the organization or the integration of the information encountered as part of consolidation. Some people say that an improvement in memory is necessary to show that there is consolidation. Consolidation is a very old concept. The first studies were done by Müller and Pilzecker already at around 1900. And they had the idea that this consolidation is actually something that stabilizes a memory. And how did they do that? They had this wonderful machine where they could present the stimuli in a very timely fashion without a computer by turning these metal tubes in this machine. And they had kind of nonsense syllables. And the task for the subject was, and they had always two subjects, Müller and Pilzecker, they investigated each other, and they had these lists of nonsense syllables, and then they varied the delay until they had to learn a second list, the list X. And then they had to a memory test on the first list, how good they were. And as you can see in this graph, depending on how long this delay was initially, there was a very high error rate for that test on list A. But as soon as a few minutes elapsed between these two, the memory or the error rate was low and flat. So then there was no interference anymore. That was actually from that paper the term consolidation came. So there's some hint of stabilization, not perhaps the most convincing, but that was at least the first one. The second point is integration. So integrating information may also enable you to make sense of complex information. And there's always that idea, if you sleep and the next morning you have the insight. And that's actually the idea that Wagner and the group of Jan Born did in this wonderful study. What they did, the subject had to learn a sequence of items. Actually, they had to learn a rule, sorry, not the sequence, a rule. So if there were two the same digits, they had to push the button of the one. If they're two different, then they had to push the button for the 9. There were only these 3 digits, 1, 4, and 9. And so on and so on. And the actual trial was correct if the last response was correct. In between was more or less, doesn't matter. But there was a trick, a shortcut. If you were correct on the second response, then you knew already that the last was the same. There was always this structure, unknown to the subjects. They had to get the insight into that. They had to get integrate the underlying structure of the task. They did many of those. And then they had 3 groups that slept normally, one that was awake during the night, and one that was awake during the day, but they learned in the morning and were tested in the evening as control groups. As you can see, only that group that slept had this insight because they were way faster in solving the task, because after the second response they knew already the final one. So there were about 60% that had that insight after a night of sleep. Being awake at night or awake during the day didn't help. So there's some insight into stabilization, integration, and now also I would like to go further into the durability of memories, the stable. Yeah.
So is the insight defined as an increase in reaction times or as
yes.
a verbal report?
No, reaction times. Then a steep decrease in reaction time. Another kind of thing that is often mixed up is the cellular and the systems consolidation. And there are some people that like to replace even the word cellular with hippocampal consolidation and systems consolidation. So intrahippocampal at the cellular level or beyond. So the cellular consolidation is really initiated after initial encoding. It's dependent on protein synthesis and appears to be happening in the first hours after encoding. The systems consolidation, in contrast, is a consequence of hippocampal neocortical interaction. And the time frame of that is, I will show you later, extremely undefined yet, indicating that there might be several processes of systems consolidation. First, I would like to bring one example of systems consolidation. I'm not an expert on that, being a human cognitive neuroscientist, but I had the privilege and the pleasure to have one joint grant with Richard Morris. And in that context, we conducted one very big animal study and one related human study, and we think that there are overlapping results in that. But we are still working on that. Richard Morris and Fry, and in this review by Redondo and Morris, formulated the so-called synaptic tagging and capturing hypothesis. What's the idea? If you have a strong memory or strong internalization or strong encoding, a tag is set in the synapse, and in the soma, proteins are synthesized that are essential to stabilize that tag, and then you have this long-lasting LTP. If you have a weak memory, then you still need the same processes, but it's a question whether you get these proteins or not. And if not, then you forget within 24 hours. And in follow-up experiments, they showed that these two processes, or these two steps, are dependent on dopamine, the release of these protein plasticity-related proteins, and on these proteins. How did they do that? They asked the animal, in this case a rat, to learn the location of one pellet in these scent wells. There were many scent wells in these boxes. And then they either wait just for 24 hours or and check the memory then, or they put the animal into a novel box that the animal was never in there to increase the novelty, and thereby dopamine is the idea. And then they test it again 24 hours later. So introducing the novel box indeed made the memory better. Without the novel box in between, the memory was at chance level, indicating that this novelty experience is actually essential to stabilize this weak memory within the first 24 hours. If they injected between the initial encoding and the novel box this sharing substance, a blocker of the dopamine receptor, this effect was gone. They replicated the novel box effect, but with the sharing blocker, it doesn't work anymore, showing that it's dopamine dependent. If they injected after the novel box a protein synthesis inhibitor, again, they replicated the novel box effect with vehicle, but with the protein synthesis inhibitor, that effect was blocked. Clearly showing that initially you need the dopamine, later you need the proteins to stabilize an initially weak memory over 24 hours. In a recent study, the same group, in particular Tominori Takeuchi, a brave, kind, and very smart postdoc in Richard Morris' lab, did over 3 or 4 years perforce through many experiments with wonderful results. And the best thing is that the results are surprising and exactly a compromise between the two hypotheses Richard and I had. I hope it's not the Japanese politeness that they come up with a compromise, but I think there was really good data supporting the idea. Richard, of course, from the data I just showed you, the VTA, dopamine, should play the trick. I have some background in emotional memory and stress, and I said, it has to be the locus coeruleus and not norepinephrine and not dopamine. So we had two opposite views on that. Now coming to the data. So first, same design as before, now with mice, because Tominori used also optogenetics, as I will see later. So this is a novelty box, pink feathers for a mice. That's something obviously very exciting. And as you can see, the memory with this novelty box is way better after 24 hours, or is the same, as good as after 1 hour. And if you inject either propranolol, a beta blocker, blocking the norepinephrine effect, or sharing the dopamine effect, Richard won, because propranolol had no effect, and sharing, as in the initial experiment, blocked the effect. So I was a bit depressed, but luckily Tominori went on. Looking into the firing rate within the VTA and the LC during the pink feather novelty box, you can see that there is a substantial increase in firing rate in the LC and a small one, but still reliable, in the VTA. So somehow the LC does something when being in the novelty box. Then the next question was, how are these two structures actually connected to the hippocampus in these mice? And as you can see, this is injecting a substance into the VTA and the LC and looking into how far that reaches the hippocampus during through the axons. And as you can see, it's not that easy to see here, but here the quantification is easy to see. The effect from the VTA is almost non-existing in the hippocampus, but from the LC it gets nicely input. So I was happy about that. Going on. So on the next experiment, as I said, they use optogenetics to activate either the VTA or the LC. And turning on the LC induced the effect I described before, but turning on the VTA increased the performance numerically, but not statistically. So 1-1, I got back. So now somehow the LC is doing the trick and less likely the VTA. In the next experiment again, they injected either a beta blocker or sharing after or while they do the optogenetics. And what you can see is that the LC on and vehicle and propranolol, nothing happens, but activating the LC and providing the sharing compound blocks the effect. So in other words, activating the LC in the novelty box releases dopamine to the hippocampus that improves and stabilizes the memory. So The LC is well known for being the source of norepinephrine in the brain, and now it looks like that it would release dopamine. However, dopamine is the precursor of norepinephrine. And in this novelty situation, the hypothesis is, and there is quite some evidence for that, that the turnover from dopamine to norepinephrine is limited, and therefore it is actually releasing dopamine. How can we translate that synaptic tagging and capture hypothesis to humans? Of course, you could do kind of sophisticated pharmacology, but we were interested in an easier way of doing it. What we did was we asked the subjects, after a defined delay, after studying object location associations, to do sport. Quite an intensive interval training at 80% of max heart rate, and then 40 hours later we tested the memory. We had 3 groups of individuals, just control. They just looked nature documentaries and didn't cycle. The one that cycled immediately after the learning and with some delay. And then 2 days later we did the test inside the MR scanner. Results are simple. The individuals with a delayed exercise had a reliably better memory than the other 2 groups. And in addition, the pattern similarity, so what Roberto today said, that if you have a more similar pattern for the successful encoding, in the left and right hippocampus for the correct trials was limited to these delayed exercise groups. So somehow the activity pattern got more similar, more clearer to the events when they were stabilized by that exercise. And exercise is well known to do many things, but of course releasing catecholamines. Yep.
So you were sure that they exercised? I mean, did we exercise in the lab?
Yes.
Yes. Otherwise they would say, yes, we exercised.
No. That was well defined on a spinning bike, and we kept them at 80% of their individual maximum heart rate.
And how long was the delay? I missed that.
Go back. So either that was immediate and that was 4 hours.
4 hours.
Yep. So much. Sorry.
No, just one more question. Why do you think that the immediate exercise doesn't help?
That's a good question. We don't know. There might many factors influence. It might either be too early, that the processes are not taken into place. Also in the rat, it's usually kind of a bit delay, or the mice. We should have perhaps chosen other delays in retrospect. It might be if we would have waited for half an hour, it would have been already in effect. And then the delayed we should have way later, that outside of the window. In retrospect, I'm not that happy about the selection of the delays. And I don't know why we did it, I have to admit. Why we choose those. Good. So much for cellular consolidation or initial hippocampal consolidation, that a memory survives at least 24 hours. The next step then is the systems consolidation. Classical view here in a figure by Paul Franklin and Bontempi. Initially, if you have a recent memory, the hippocampus has the connections to neurocortical modules that represent the features of the memory. And with time, these connections of the hippocampus to the neocortex get weaker, and the connections between the modules in the neocortex get stronger. And after a full consolidation, you're able to retrieve your memories without having the hippocampus. Classical finding of the temporal gradient in medial temporal lobe retrograde amnesia. But how long is this gradient? Oh, how steep is the gradient? I summarized just data from one group. One of the most influential and respected neuroscientists of memory, Larry Squire. So here he asked people, either control subjects or patients with hippocampal lesions bilaterally, to recall news items that were published either 5, 10, 15, 20, or 25 or 30 years before the investigation. And as you can see, 5 years perhaps 10 years, the control subjects were substantially better than the hippocampal amnesia patients, indicating after about 10 years, memories are hippocampally independent. If you ask for childhood memories in a systematic way, you can see that they're almost perfect, even with hippocampal and myotemporal lobe lesions. Only those patients with very extended lesions due to herpes encephalitis have even damage to these childhood memories. Going on with electroconvulsive therapy, something that interrupts this process. Patients were tested in 1974, and as you can see, if the investigation, if they tested memories or famous faces a few years before, then, or known a few years before, there was a big difference before and after ECT within subject comparison. But already kind of 4 or 5 years, no difference anymore, indicating 4 or 5 years, memories are hippocampally independent, already 50% reduction. Again, the same group, now monkeys, prospective study. Monkeys learned, and at different intervals in different animals, of course, after learning, the hippocampi were removed. If you remove the hippocampus 2, 4, or 8 weeks after learning, you have quite a nice difference between those animals that got the hippocampi removed as compared to the sham-operated animals. But after around 10 weeks, there was no difference anymore. So reducing the hippocampal dependency to 10 weeks now. Now I would like to make a short detour because it's relevant for the rest of the lecture. It's about the role of sleep. I mentioned already the effect on sleep on the INSIGHT study, but I would like to go a bit further. Sleep is not kind of a stable state. It had different stages, and the most interesting for this lecture is the slow wave sleep, the delta sleep, but otherwise from drowsy sleep stage 1, 2, and the rapid eye movement sleep stage. These stages are not evenly distributed over the night. You have usually 90-minute cycles. When you go to bed, you have immediately a stage where it goes down to deep sleep, and you cycle in a deep sleep, and towards the morning, you have more REM sleeps. Stages are not that much deep sleep anymore. And again, the group of Jan Born showed several studies. Very nice one is a perceptual task, just a perceptual skill task. We have to identify the odd white bars there when fixating here. And as you can see, if the subject slept for 8 hours or already for 3 a bit, but when they stayed awake during the day or during the night, there was no improvement, but otherwise there was improvement. Here is really an improvement in memory. What you can see is they learned prior to sleep, then they slept, and after sleep, they didn't continue at that level. There was a leap up, and they performed better. This is one of the few examples in this skill learning where the people are really getting better with memory.
Sorry?
Yep.
Isn't this an example of perceptual learning?
It's perceptual learning. Yeah.
Is it sleep dependent, like more high-level tasks?
Yeah. Here they have 2 different types of memory, and this gets even more specific to different stages of sleep. They have, as I told you, they're in the first phase or first half of night, you have quite some deep sleep. Second half, you have quite some REM sleep. And people were woken up in the middle of the night. And either they learned and retrieved in this way or learned and retrieved in that way. 2 different groups. And they had 2 different types of memory, pair-associated episodic memory and mirror tracing in a skill learning trial. And as you can see that the early sleep, or the early sleep, the slow wave sleep, was particularly important for the episodic memory, while the REM sleep was more relevant for the skill learning. What is happening during sleep? There are quite some ideas. The most prevalent one in the field is, of course, the idea of replay. Here's a nice picture from Atherton and colleagues in a review. What replay says is that if a rat has visited these dots in a maze and in a certain order, then these place cells are firing in a certain sequence, and during the sleep, this sequence is replayed. And there, people like McNaughton, Wilson, and others have repeatedly shown the role of these things. How can we do that in humans? And again, Jan Born, Björn Rush as the first author, did the first study with acute reactivation during sleep. What did they do? They had to learn object location associations, and for each during the learning, they had a rose smell in the room or nothing. And then they went into sleep, and when they were in deep sleep, they presented again the smell to the subjects. And then on the next day, they tested them. They had 4 groups: the odor or vehicle during slow wave sleep, and then the test next morning. No odor during learning, but odor during the deep sleep. Third group, odor during learning, but odor or vehicle during REM sleep, or odor during learning and odor or vehicle during awake at the same time in the evening, and then the test. So nicely showing that only the group that had odor during learning and slow wave sleep had a substantial improved memory for the object location associations, all others not. Moreover, they did an MRI study where they scanned with fMRI during slow wave sleep and presented blocks of rose smell or nothing during the sleep. So smell on, off, on, off. And as you can see, the presentation of the odor activates the hippocampus in slow wave sleep. And it was again related to the better memory. But of course, it was the entire kind of learning episode poured into a smell. This was not specific. Therefore, Ken Puller and his colleagues, Mark Rudoy as the first author, did a more specific study, very similar, but now no smell was presented, but sounds. So to each object that had to be learned in the location, a specific sound was presented. Meow for a cat, a whistle for a kettle. And during slow wave sleep, only 50% of those sounds were presented. Therefore, you have cued and uncued sounds during the night. And as you can see, the uncued are way better. This error. The cued were way better than the uncued. So if the cue was presented during slow wave sleep, then you were able to remember the location of the object better. So that's an indication of an induced replay. We did the same study in collaboration with Ken Puller and Mike Rudoy in the fMRI. This is a very painful design. Never think of doing that, because you have to get your subjects into the scanner, sleeping into deep sleep, and present stimuli without acoustic stimuli without waking them up. And that is prone to fail. So to get a group of 20-something subjects, you have to scan about 70 people, all at night. So don't do. Elke van Dongen, excellent postdoc, or at that time PhD student, left science. He's now working at the Dutch Science Foundation. So what did he do? Again, the same task. You have to learn the location of the object, presented with this associated sound. They went, and we did that already in the fMRI, then they went into the scanner for the night. And during slow wave sleep, so you monitor continuously the sleep stages of the subject, and if they are stable slow wave sleep, you start the presentation of the stimuli, of the acoustic cues, only 50%. And then, and the next morning, you do the test. So, about fMRI during slow wave sleep. So during slow wave sleep, if you just contrast the activity of the cue sounds compared to a controlled sound, a matched controlled sound, you get activity in the posterior parahippocampal cortex. So somehow the brain is processing the sound differently whether you have learned them or heard them shortly before scanning as compared to other. If you put a seed region in this activity difference, then you see that this region has a differential connectivity pattern to several areas that are important for the representation of objects and locations. So somehow the entire network gets connected and activated during slow wave sleep if you present these cue sounds during slow wave sleep.
What happens if you wake up the subject and present the cue sound?
This was done before by Ruda. We didn't do that here.
But you get the same effect?
I don't think so. No. So for all the other experiments, not.
You don't think you would in this one?
No. If you go to activity that's cue related that predicts whether you're able to remember a specific event later on, that's kind of a cue related subsequent memory effect during slow wave sleep, you get activity in the thalamus and the hippocampus. So there is activity that predicts whether you're able to remember, or how well you're able to remember. It's not whether, it's always location. You remember all, but it's just location. How well. And during the post-sleep test, the connectivity of the parahippocampal region I told you with this myoprefrontal region is increased depending on performance. And this myoprefrontal cortex activity will come back in my lecture. What's the time again?
I never know which is which, but I guess you have 5 minutes or so.
Yeah.
Okay. Okay. Good. I will be almost done with consolidation. Yeah.
Can I just ask about that region?
Yes.
So that's what we call pregenual cingulate cortex.
I agree. That is hard. It goes further than that. And I agree with you that the localization is difficult. But that's what that means in terms of decision making, I'm not sure.
Rewards are
Reward.
represented there.
Yeah, will be hard for reward, I think. Let's see. Another study also by Elke van Dongen, so he got into the sleep study. And this is a simpler study, just a nap. And what you see is that they learned prior to the rest or the nap, and then they learned a second list, and then you have items that had been processed or consolidated during that nap, and items that you don't. Very simple. And during this nap, the subject went into deep sleep. But nevertheless, we were able to measure the connectivity of the fusiform gyrus. Why the fusiform gyrus? In that test, we looked for phase location associations. Fusiform gyrus is important for phase representation. And therefore, we put a seed region in the fusiform gyrus and looked how that was connected during shallow sleep or middle stage sleep. And as you can see, in particular the slow wave in this shallow sleep, that the entire dorsal and ventral visual stream was more connected to the fusiform gyrus as compared to wake. So somehow already at that stage, the activity is increasing in a certain network. And furthermore, there was also an activity or the connectivity with the fusiform gyrus, and again, anterior. Pregenual, okay. ACC shows a predictive connectivity to the ability to remember after the rest. And also the medial-temporal lobe connectivity with the fusiform gyrus predicts the acquisition of new items after the rest. This one is particularly interesting because here again it's very similar, it's almost the same task, contrasting remote and recent activity during retrieval, and you get again this mere prefrontal as I called. So somehow we have some evidence that we kind of, or actually not that much evidence, but that is actually I should do that after the lecture. That's the next point. So far I presented data showing how the process initially, how encoding is happening and what processes at the system level are occurring there in the first 2 seconds, then what might happen during the first 24 hours with the one spotlight on the tagging and capture hypothesis, and then initial ideas about replay during sleep that might stabilize the memory for later retrieval. And thereafter I will go on with that story.
Great.